How's the studying going? Quiet group. Okay. So we'll do these like I've done the other ones. So I'm here for your questions. So whenever you have questions, raise your hand. So I'm guessing the questioners were the ones who didn't come, and then these are the ones that we're going to, this is the group that came to listen to the questions. Is that right? So let me check the camera, make sure everything's okay with it while you're thinking of your questions. So just to say a little bit, like I said in class, the exam will have 150 points, same format as you've seen before. The final tends to be a little bit shorter because I tend to make a few more questions on the short answer than I do on the problems. And that's partly because we have to grade this by Monday. And that's usually a challenge to get together since we don't get to start grading it until 8 o'clock on Thursday night. So, but that's, I haven't written the exam yet. In fact, I haven't even thought about the exam yet, other than the fact I'll make it as hard as I possibly can. So that's my plan. Yes, a question, a brave soul.
I do have a question. The last video we went over, which was maybe enzymatic activity with the addition of glucose.
Yeah.
Could you repeat that?
Yeah, sure. That's a really cool figure. So what she's asking about is this figure that has glycogen synthase, glycogen phosphorylase, and they add glucose to it. And what happens? And glycogen. And what they see is this. Okay? So what this tells us, if we were just to look at this in the absence of everything else, what this tells us is that glucose has an effect. Now, you know glucose should have an effect on the glycogen phosphorylase. Because if you recall, glucose, when the R form of glycogen phosphorylase is present, glucose is an allosteric regulator. It's a negative regulator of glycogen phosphorylase and causes it to flip from the R into the T form. So it's not totally surprising that you should see there would be a decrease in activity of glycogen phosphorylase. However, what you see is not only a decrease in activity, which is measured here, but you also see the form change. You see it change from phosphorylase A down to phosphorylase B. Okay? Well, that means there has to be a dephosphorylation going on. And in addition, you see glycogen synthase is changing from the synthase B into the synthase A. And that also happens only as a result of dephosphorylation going on. So what it tells us is that there has to be a phosphatase present. There's just no way there's not a phosphatase present. Phosphate doesn't just fall off of things. Something has to be there. So when you have glycogen phosphorylase and synthase present in the presence of glucose, you have a phosphatase. Well, that was the first clue that there was something going on with that, and that is actually shown in this slide right here. It turns out that the phosphatase is actually linked to the glycogen phosphorylase A in the R state. When that happens, we have this configuration that you can see on the left. That is that this protein G, this is the liver form of it called G/L. The other one I showed you before was G/M, which was from muscle. But this G/L has a binding site for glycogen phosphorylase A in the R state. And this phosphorylase A blocks the catalytic site of phosphoprotein phosphatase. So in this circumstance, we have and remember that also in this reaction, we also have glycogen synthase in the phosphorylated form. So we're starting with a phosphorylated form of glycogen phosphorylase and a phosphorylated form of glycogen synthase. So the addition of glucose causes these two things to happen here. First, the addition of glucose binds, the glucose that's added binds to the phosphorylase A in the R state and converts it to the T state. And that conversion of phosphorylase A from the R state to the T state is sufficient for it to be released from the phosphoprotein phosphatase and GL complex. So now we have glycogen phosphorylase A in the T state, okay? And the phosphatase is active. Well, the phosphatase, of course, can take phosphates off of any protein. So it takes phosphates off of phosphorylase A, which converts it to phosphorylase B. And it takes phosphates off of the glycogen synthase B and converts it into synthase A. So that has the effect of essentially knocking out glycogen phosphorylase from activity and activating glycogen synthase activity. Now, as I said in class, the importance of this is the fact that this is a very rapid change. Once glucose starts to accumulate in a cell that is breaking down glycogen, then that cell is very rapidly converted into not breaking down glycogen and starting the synthesis of glycogen. And again, when you have abundant glucose, that's what you want to have happen. So that's what's really going on in this set of reactions. That answer your question? Yes, Justin.
Building off of kind of glucose metabolism, could you go over again how ingestion of fructose skips regulatory steps?
Okay. How does the ingestion of fructose skip regulatory steps? Okay. So let's go back to our discussion of glycolysis and talk first of all about fructose metabolism. All right. So fructose metabolism is shown right here. And fructose, of course, the body has a variety of sources of fructose. Fructose comes from the breakdown of sucrose, which contains fructose and glucose. And many fruit juices and other things have fructose in them. There's quite a wide variety of ways in which you can get fructose in your cells. So fructose is metabolized as you see here. That is, fructose is converted first to fructose 1-phosphate by this enzyme known as fructokinase. And fructokinase makes fructose 1-phosphate, which then gets cleaved by this enzyme fructose 1-phosphate aldolase. And this is actually quite easy to remember because aldolase the name aldolase in the glycolysis pathway breaks down fructose 1,6-bisphosphate into DHAP and GAP. So this aldolase breaks fructose 1-phosphate into DHAP also, but then also into glyceraldehyde. Glyceraldehyde, of course, doesn't have phosphate here. That phosphate is readily put on by this enzyme known as triose kinase, and so we have glyceraldehyde 3-phosphate. So if we look at the products of this reaction, we see that we have two glycolysis intermediates, and we made those two glycolysis intermediates without going through the regulatory steps shown here. Okay? So PFK is a regulated enzyme, and that PFK will respond to cellular needs for energy and so forth. And so it's actually a good stopping point to stop the production of fructose 1,6-bisphosphate and thus stop the rest of the pathway when the cell's needs dictate that they don't need all that stuff. The problem with fructose metabolism is that fructose metabolism bypasses PFK. Remember that fructose is coming in as fructose 1-phosphate, and it's getting broken down into DHAP and glyceraldehyde, and so it's actually entering down here. Well, the effect of that is to basically take all the carbons that are in fructose and make them enter glycolysis, but the only regulatory enzyme that they hit is pyruvate kinase. Okay? And pyruvate kinase, if it is at all active, will convert phosphoenol pyruvate into pyruvate. So what that means is that fructose metabolism is going to feed the production of pyruvate, And pyruvate, as I noted in class, is a precursor of acetyl-CoA, which is itself a precursor of fatty acids. So it's through that alternate pathway that causes, I think, and this is again my idea, but this is why I think that we can blame high fructose corn syrups partly on the obesity epidemic. Now, in reality, people say, well, people have been eating sucrose forever, and sucrose has fructose and glucose, so aren't you normally doing that if you're getting sucrose? And to that I say, yeah, probably. The more carbohydrate you put into your diet, probably the worse off you are. And for the reason I've stated here, but also you may actually be forcing the production of pyruvate through this pathway as well. And so in both cases, you're going to make something that's an intermediate in the synthesis of fatty acids, and that's not a good thing at all. Clear as mud? If you want to lose weight, so if you want to lose weight, what you have to do is you have to burn more than you eat. It's very simple. Okay? It's very, very simple. Okay? There's no magic bullets. There's no magic pills. There's nothing more than burning more than you eat. And that will cause you to lose weight. Yes?
If you're diabetic with low blood sugar,
Yeah.
would it be better to have fructose or glucose?
So if you're diabetic and you have low blood sugar, it would be better for you to have glucose or fructose. I'm not allowed to give medical advice. I'm sorry. And I will not do that because I don't want to be sued. But thank you. Yes? The what? Oh, yeah, sure. The regulation of the PFK-2? Okay, sure. The most hated figure in the entire term. And that most hated figure is right here. Okay? This is the one you're talking about, right? Okay. Everybody's shaking their head. Yeah, this is the most hated figure, right? Okay. Well, before I start talking about it, I'm happy to, and I'll lead you through it. But before I start talking about it, I really strongly encourage you to sit down and write this thing out. And I think when you do that, you're just going to see that I can throw, 1,000 words at this thing, and it's going to be a waste of words compared to what you write down. All right? I'm happy to give you those words, but at the same time, if you sit down and you write this thing down, you're going to see it's really not as bad as the words sound. Okay. So we start with this thing, recognizing what the overall purpose of this pathway is. This pathway that you see on the screen is for the synthesis and breakdown of fructose 2,6-bisphosphate. That's what it's there for. Synthesis and breakdown of fructose 2,6-bisphosphate. Okay? The synthesis is shown on the left. You see the synthesis of fructose 2,6-bisphosphate. On the right, you see the breakdown of fructose 2,6-bisphosphate. Okay? So if we have a way to make a compound, we have a way to break down a compound. Making a compound that has phosphate, we're going to use a kinase. And breaking down a compound that has a phosphate that we don't want, we're going to use a phosphatase. Okay? All right. So that's actually what's in here. So the kinase and the phosphatase are both pieces of the same protein. They're both pieces of the same protein. The kinase part we call PFK-2. The phosphatase part we call FBPase-2. Okay? And as you can see in the figure, only one part of that is active at any given time. Only one part is active. So either the kinase is active or the phosphatase is active, but both are not active at the same time. Okay? If the kinase is active, we're going to make fructose 2,6-bisphosphate. And fructose 2,6-bisphosphate will stimulate PFK, and stimulating PFK will stimulate glycolysis. If the phosphatase is active, we will break down F26BP. And breaking down F26BP has the opposite effect. It stops glycolysis, and it stimulates the gluconeogenesis. Okay? So we have exactly opposite things going on. We have a very nice reciprocal regulation. The question then is, well, what causes the enzyme to be in either state? The phosphorylated form of the enzyme is the phosphatase active form. The dephosphorylated form of the enzyme is the phosphatase inactive and kinase active form. Okay? So I put the phosphate onto the protein when epinephrine is stimulating a cell. Whenever you see protein kinase A being active, that means epinephrine has bound to the cell surface receptor and has stimulated the activation of protein kinase A. Putting the phosphate onto the PFK-2/FBPase2 complex causes a phosphate to be put on. FBPase2 is active. That means fructose 2,6-bisphosphate will be broken down. And when we break that down, it means glucose is scarce, so we're going to turn on gluconeogenesis. Okay? And if you think about this, this makes sense because epinephrine was being produced when the body needed energy. The liver is a major source of glucose. So what you're seeing here is going on in the liver because in the liver, two things are happening. One is glycogen will be broken down, and the other is glucose will be synthesized, both of which increase the concentration of glucose in the bloodstream. On the other hand, if blood glucose levels get high, then insulin is produced in the body, and what insulin stimulates is the activation of a phosphoprotein phosphatase. Phosphoprotein phosphatase takes the phosphate off of this complex, converts it back into the form where PFK-2 is active. That is, the kinase is active. And when the kinase is active, we phosphorylate fructose 6-phosphate to make F26BP. And F26BP stimulates PFK. It inhibits gluconeogenesis. And we favor the breakdown of glucose. And this also makes good sense because when glucose concentrations are high, we want to reduce the concentration of glucose. And two ways to do that. One is by the breakdown of glucose, and the other is by the synthesis of glycogen. So insulin stimulation will favor both of those processes. Long answer to a short question. Does that help clarify anything for you? A lot of words, but the figure, I think, says it much more efficiently than my words say it. Yes, sir. Yes? Okay. Triose phosphatase is an enzyme that I described as a perfect enzyme. And the question is, what about triose phosphatase keeps the undesirable intermediate from forming? So that's actually shown right here. And more specifically, it's shown right here. So if we look, and I didn't talk about this in class much, but I'll briefly mention it here for you. There's an enediol intermediate right here, which is an intermediate. And this intermediate turns out to be unstable. Okay? So this intermediate is chemically unstable, and if it's allowed to sit around for any period of time, what will happen is it will convert into methylglyoxal, and that conversion happens without the enzyme. It'll happen chemically, and methylglyoxal is poisonous. Okay? So the cell doesn't want to have methylglyoxal around. It wants to have glycolysis intermediates around. So one of the reasons that the enzyme is perfect is so that this enediol intermediate, as soon as it forms, is rapidly converted into a form that is not unstable. So that the way that the enzyme is helping the cell out is by reducing the likelihood of the formation of methylglyoxal by having an enzyme that is extremely efficient. Now your question was, how does it do that? And it does it simply with speed. It simply does this reaction extraordinarily rapidly and prevents this from sitting around for any period of time. Okay? It's a pretty simple mechanism, but it's kind of a cool way to do things. And this perfect enzyme strategy is remarkable to me to think about, that there's actually a possibility of making a perfect enzyme, but then also to see that nature appears to have actually managed to do it, which is pretty cool. should slow down. You're still typing after I shut up. Now you got to type that too, right? I will. The Cory cycle, yes, sure. So the Cory cycle is one of my favorite cycles. I think it's the first example that you see in the body of yeah, I hate that figure. It's the first example you see in the body of basically metabolic pathways helping each other out. So I'll start with the Cory cycle by saying a little bit about it that sometimes students find confusing. And so I talked about reciprocal regulation. And reciprocal regulation, you remember that the principle of reciprocal regulation is that you don't want to have catabolic and anabolic processes going on at the same time, because you'll make a futile cycle where you have one breaking down and then the products of that becoming the synthesis for the other, and the breakdown and the synthesis and the breakdown and the synthesis, and you have a problem. And so on the surface, the Cory cycle looks like it's a it's a futile cycle, because on the one hand you are making glucose, and on the other hand you're breaking glucose down. But futile cycles are only futile when they occur in the same cell. And so what's happening in the Cory cycle is you have specialization. This is a really good example of specialization happening in the body. The liver cells are specialized to produce glucose for the muscles when they need it badly. And so in the liver, what we see going on is the synthesis of glucose. The starting material for that is lactate, as you can see here. And lactate, of course, comes from the muscle cells. But there's no breakdown, there's no significant breakdown of glucose that's occurring in these liver cells. They are simply converting lactate into glucose. So this is not futile here. By dumping the glucose out of the liver cell and into the bloodstream, what the body is doing is basically getting rid of excess glucose in the liver and giving it to the muscles that need it badly. The bloodstream delivers it to the muscles, which grab the glucose and just take it up like crazy. And one of the real advantages for glucose, and I mentioned this briefly in class, but I'll mention it here again, is that glucose is soluble in the liquid of the blood. Okay? When you talk about fatty acid metabolism and fat metabolism next term, you'll discover that they're not soluble in the liquid of the blood, and they have to be packaged. And so that takes time, and it's a relatively inefficient process. Okay? So that means, therefore, that energy from fat and fatty acids is not a very good source or a very good way of filling the body's immediate needs. But putting this into the bloodstream where it dissolves and just travels very quickly is a very, very fast and efficient way to feed muscle cells. So muscle cells get this dissolved glucose in them. They take it in and break glucose down, and they're breaking it down to lactate because a rapidly exercising muscle is using oxygen faster than the bloodstream can deliver it. And that's entirely possible for you to do. When that happens, of course, we have fermentation going on. So instead of having glucose going solely to pyruvate, pyruvate gets converted to lactate to keep glycolysis going, you may remember. And then lactate, which is the end product of this pathway, is not needed by the muscle cell. It dumps it out into the bloodstream. And lactate is also soluble in the liquid of the blood. And that ends up going back to the liver. So we have this cycle that's going on. And though it looks futile, it's not futile because it's occurring, the synthesis part is occurring in different cells than the breakdown part. Make sense? Excuse me. Justin.
I know that lactate is used in gluconeogenesis as a precursor molecule.
It is a precursor molecule, huh? Yeah.
Okay. And you also mentioned the conversion of, I believe it was lactate to acetic acid.
So the question is, I talked about, so he's asking about the fates of lactate. No, I did not talk about lactate going to acetic acid. I talked about pyruvate going to acetaldehyde and acetaldehyde can go to acetic acid. Okay, so but I did not talk about lactate. Lactate's pretty much a biological dead end. About the only thing you can do with lactate is convert it back into pyruvate. And that's actually what's going on right here. So cells that are rich in oxygen will convert lactate back to pyruvate. Okay? And pyruvate can be used to make acetyl-CoA, and acetyl-CoA can be oxidized in the citric acid cycle. Now, it's interesting, and I actually learned this from a student in the class a few years ago. I always like to learn from my students where I can, and this was a cool thing that I learned, and that is that lactate is actually a pretty good energy source for the heart. Lactate's a good energy, so you don't need to know this, but I'll tell you briefly. You might think, well, how is it that lactate is a good energy source for the heart? The heart, of course, is right next to the lungs, and the heart gets oxygenated blood very readily. And what the heart does with that oxygenated blood is it converts lactate into pyruvate. It converts pyruvate into acetyl-CoA, and acetyl-CoA is oxidized in the citric acid cycle, which is a very abundant source of ATP. So if you think about this, this balance in the body is really cool, because here are muscle cells that are out, they're exerting, they're going like crazy, they're producing a lot of lactate, and what are they feeding? The heart. Really cool. Really, really cool thing. So, pretty neat. I hope that when you guys are done with metabolism, that you can run these pathways through in your head like I do. Because when you think about these and you can understand how the body works and how cells work, metabolism is a very, very powerful tool. And you can understand a lot of things about nutrition. A lot of students, I take my 350 class who are nutrition majors and find that metabolism really is a lot more interesting than what they thought it was. It wasn't just a matter of memorizing pathways, although you have to know pathways in order to understand some of these things. But it tells you a lot about how the body works and what the different regulations of those pathways are. So. So let me ask you guys a question. I'll ask you a question. And I'm not going to use this information against you, okay? So my question, I'm just curious. What do you feel has been the most complicated thing of the term? What has been the most complicated?
I would say understanding all of the regulatory steps within glycolysis and gluconeogenesis.
Okay. The complexity of that?
Yeah.
Okay.
How each different molecule affects different enzymes, and then how that affects regulation of either glucose synthesis or breakdown.
Okay. How many people agree with that? That the regulation in glycolysis, gluconeogenesis was the most complex? Okay. Interesting. Yeah. Yeah. Yep. And you're going to see a lot more of that next term. Because when you start overlaying, that's the good news, guys. When you start overlaying fatty acid metabolism and the citric acid cycle and electron transport and oxidative phosphorylation on top of this, you really get a cool picture. You're going to learn what's called respiratory control. And you're starting to see a little bit of that here. The Cory Cycle is a little bit of an example of a respiratory control, very simple kind of respiratory control that's happening in the body. But you'll see that with respect to electron transport and oxidative phosphorylation. And these processes actually are completely fed by the metabolic end products. And so there's a very definite link with all of those. So I'm really missing not teaching this next term, I will tell you that. I find that, as I said, this is the first term in 12 years that I haven't taught the two together. And it's just a natural pairing. It just really, kind of regretting not teaching it, but too late for that. And I'd say the other thing about it is you guys have been a very, very good class. That I've had some classes, and I'm not just saying this in front of you guys, but I've had some classes where sometimes you just feel that people don't really give a shit, you know. This class has not been that way, so I've really enjoyed it. Yes, sir. HIF? You're talking about HIF1, the hypoxia induction factor? Okay. Okay. So hypoxia induction factor, which is, let me think where that is. That's actually not here. That's before here. Here? No. Previous to that, it would be here. No, it would be previous to that. Well, maybe not. Where am I at? It's what? It's where? 24th of what? November. Okay. All right, so then it is later then. Okay. It all runs together to me after a while, I'll be honest with you. So, well, where is it? Say where? Oh, here it is, right here, right here. Okay. So, this is what you're talking about. Yeah. Okay. So, again, coming back to metabolic control, you've seen how metabolic processes are most efficient when oxygen is most abundant. 38 ATPs in glycolysis, from glycolysis, not in glycolysis, but from glycolysis compared to 2, oxygen present versus oxygen absent. Okay. So oxygen is a major consideration for cells, and cells have to be able to adapt to conditions where they have reduced oxygen, because there are times when cells are going to have reduced oxygen. That's just a function of exercise and the fact that the delivery systems that we have are not ideal, or I shouldn't say not ideal, but not 100% efficient. So we can exercise faster than we can deliver the things that we need. Well, the HIF1 is a protein that is a reflection of that. HIF1 stands for hypoxia induction factor, and HIF1 is something we call a transcription factor. Okay? A transcription factor. So a transcription factor you'll learn a little bit more about next term, but a transcription factor is a protein that binds to a specific sequence in DNA and causes the genes near that sequence to be transcribed. Okay, so transcription, of course, means the synthesis of messenger RNA. Synthesis of messenger RNA leads to synthesis of protein. So what HIF1 is providing is a way of turning on synthesis of proteins that are needed, and in this case, they're needed when oxygen is of low abundance. Okay? So when oxygen is of low abundance, this is the condition known as hypoxia, HIF1 will be activated, and the activation of HIF1 causes it to bind to DNA and turn on these genes that are needed when oxygen abundance is low. Right? Well, so there are several places where oxygen abundance may be low. They may be low in a heavy exertion, as we've already talked about. When I'm out jogging like I was this morning, okay, my muscle cells were running low on oxygen. I probably was inducing activation of some HIF1 as a result of that. And those cells that were getting the HIF1 activated were having some enzymes being synthesized as a result of that. The other way in which this can happen is with a tumor cell. And tumor cells are, I shouldn't say all tumor cells, but many tumor cells are naturally what we call hypoxic. And that's because, first of all, they're undifferentiated tissues, so they tend not to have blood supply going through them. Many tumor cells will synthesize hormones that actually stimulate blood vessel growth, which helps to feed them. So successful tumors are those that can induce the body to give them blood. And successful tumors are also those that can adapt to those conditions of low oxygen. Okay? So in a tumor cell, it's not uncommon to see HIF-1 has been activated because they're already low on the oxygen scale. Well, with that all said, the question then is, what are the proteins that are synthesized? And this is a list of some of the proteins that are synthesized by an active HIF-1. And so these first two proteins are called GLUTs that I talked about earlier. You'll talk about GLUTs a little bit next term also. GLUTs are glucose transport proteins. They, you may remember, are the way that cells bring in glucose from the exterior. You remember that insulin stimulation favored the movement of GLUTs to the cell membrane so that they could open up and let glucose come in. So if you have a circumstance where a cell is low in oxygen, then you want to get that cell as much glucose as you possibly can. So making GLUTs to bring in glucose is important because remember, again, I know I keep repeating it, but remember that this cell that's lacking oxygen is going to need 19 times as much glucose as a cell that has oxygen. So having having GLUTs to handle that and bring that in is really, really important for the survival of that cell. Okay? Well, once you get the glucose into the cell, glucose, of course, is a poison. So if you don't handle that poison, what's going to happen? Well, you're going to kill the cell, and that's not very good for, from a cellular perspective for survival. So having a way to handle that glucose is important. And so if you bring in more glucose, it's important to have more enzymes to handle that glucose. And so these enzymes synthesis are all induced when HIF-1 is activated. And this allows now the cell to bring in glucose and then to handle all that glucose that it has inside of it. And as a consequence, in our body, normally we have cells that are needing this. They're functioning okay. But unfortunately, also, this is also helping cancer cells to survive as well. There are some strategies, and many of you have asked me about them, and I'll repeat what I've said before. There are some strategies for killing cancer cells by identifying those which are hypoxic and then starving them as much as possible. There are some strategies that block the action of blood vessel hormones so that the tumor cells which are secreting this hormone to make blood vessels have the hormone blocked, and as a consequence they get less blood supply and are much more treatable than those that don't have that hormone blocked. That's one strategy, and there are others as well. Simple question, long answer. Justin?
I know that UDP glucose plays a role in galactose metabolism.
Mm-hmm.
And is that due to the energy held within the bonds of the activated intermediate? Does that help to metabolize glucose or galactose?
Okay, so the question is that UDP glucose plays a role in galactose metabolism, and UDP glucose is an activated intermediate. Does the energy of that activated intermediate affect galactose metabolism? And the answer is no, but I'll show you why that's the case. So if we look at galactose metabolism, we see this guy right here, this figure that I showed you before. If you look at what's happening in this figure, all that you're doing is breaking a bond, a high-energy bond, and then replacing it with a different one. So it's not like there's any net energy that's realized as a result of the breaking of this bond. So here's galactose 1-phosphate. This high-energy bond's being broken, but it's simply the energy is simply being used to connect the UDP to galactose. Okay? This produces glucose 1-phosphate, which goes off and does its own thing. Okay? This UDP galactose is epimerized, meaning that an epimer is made, and an epimer changes the configuration of one hydroxyl. There's the hydroxyl that's being changed. And when you've changed that, you've got UDP glucose. But you notice we still have a bond that's occurring. That is, we still have that high energy bond there. So there's been no change at all in the bond that's happened with that. So the fact that it's working with an activated intermediate really has no effect whatsoever on the metabolism of this thing, because there's no use of the energy of that high energy bond.
Okay, so essentially it's just converting galactose into G1P so that it can enter glycolysis, which is amino acids.
That's correct. So what he said was that the process is converting galactose 1-phosphate into glucose 1-phosphate so it can be used in energy production, and that's exactly what's happening. Yes?
So you mentioned debranching enzyme and the breakdown of glycogen.
Mm-hmm.
And there were two parts of it. There was the transferase and there was the alpha 1 glucosidase. Do we know as separate parts?
No.
Or just debranching enzyme?
Right. So the question was that the debranching enzyme has two activities that were shown in class, and I'll show you the figure that has those activities. That actually is right here. Okay. And the question was, do you need to know both these activities? And the answer is no, you don't. Okay. You should certainly know what the enzyme is doing, which is debranching it and breaking off glucose, but you don't even know the names of these or anything like that. No. No. I like to keep this. And I think that, as I said in class, glycogen metabolism is very simple. And the simpler we can keep it, I think the better. So, how are your cards coming along? There's people shaking heads. What's that? So is there a strategy of wait till the last minute to put down the things that you don't know, or are you making them as you're going along, or what are you doing? As you go along, okay. I think most people actually wait till the end. Yeah.
I started writing on it yesterday just to make myself start filling it out as I study.
It's interesting that the effect that the card has. I don't change the exams that I write. You'll see the exam will look just very much like the exams you've already seen. But it has very little effect on the grades. The averages don't really vary because of the card. I think what the card does is it gives you security. It's a security blanket. You feel good having that card there, which I think is kind of cool. Yes? Say it again. Uh-huh. I'm not sure what you're saying, but I'll just say a little bit more about what's up here. Two main things up here. One is that the glycogen phosphorylase can only work to within about 4 or 5 residues of a branch, and that's what's shown right here. So the glycogen phosphorylase will break the reds out of there, and that's why you see these 1, 2, 3, 4, 5, 6, 7, 8. You see 8 glucose 1-phosphates that are produced by that phosphorylasis reaction. And then this structure has to be resolved, and that's where the debranching enzyme comes in, moving these 3 to the end and then clipping the glucose off of that one. But I'm not sure if I'm answering your question. Okay. Justin?
I think this has a little more to do with what we'll be learning next term in the cycle.
Okay.
But do you go over how citrate affects PFK and fructose corn phosphates?
Okay. So how does citrate affect the glycolysis gluconeogenesis enzymes is basically what you're asking. And since you've asked me for it, I will say a little bit about it. And I will say a little bit about it relative to next term, but you won't be responsible for that part of it, okay? So you should know something about this regulation, the reciprocal regulation of these pathways, okay? So this figure shows it pretty nicely. The most important one, as I noted, is F26BP. And the reason it's the most important one is because it takes very, very, very tiny amounts to affect these two pathways. Very tiny amounts of that molecule turn off the fructose 1,6-bisphosphatase and turn on PFK. Very, very tiny amounts. In fact, there's such tiny amounts of this that have this effect that this molecule wasn't even detected until about the 1980s. Okay? So very, very vanishingly tiny amounts of this molecule that have this effect that's on these enzymes. What you asked about was citrate, not F26BP. But there are three things that you can see here that are reciprocal regulators of the enzyme that catalyzes reactions between these molecules. PFK going down, fructose 1,6-bisphosphatase going up. And the three molecules are F26BP, AMP, and citrate. And they have exactly the opposite effects on the enzymes. F26BP is not anything relating to energy. The other two have something to do with energy, but F26BP is simply an indicator. You can think about it a little bit like a signaling molecule within a cell, if you want to. Okay? We think about signaling molecules between cells, but the signaling molecule within a cell is really what F26BP is here. You saw the elaborate way in which its synthesis and breakdown is controlled. Okay? But it has nothing to do with energy. It has to do with what which hormone is hitting the cell. That's really what it has to do with. All right. AMP, on the other hand, is an indicator of energy. Okay? So AMP is produced when the cell has low energy. And when you have low energy, you want to burn glucose to make ATP. No ifs, ands, ors, or buts about it. And so you want glycolysis to be going, and you don't want gluconeogenesis to be going, because gluconeogenesis will require energy, and you don't want to be making glucose in a cell that already is low in energy. So AMP turns on PFK, turns off fructose 1,6-bisphosphatase. Citrate, which you asked about, is also an indicator of energy. Okay? Now I'm going to tell you why it's an indicator of energy, but you won't be responsible for knowing the why about why it's that. Okay? When you talk about the citric acid cycle next term, you'll see that you take a 4-carbon intermediate known as oxaloacetate and a 2-carbon intermediate known as acetyl-CoA, and you put the two together, you split out the CoA, and you get citrate. Okay? And citrate, 2 steps later, is oxidized. Okay? So citrate gets converted into isocitrate, which itself gets oxidized. And oxidation requires NAD, which means it requires oxygen, and so forth. If you are rapidly burning things, you have plenty of oxygen supply, what's happening is your citric acid cycle is just going through the circle, and it's a circular pathway, just going on and on and on and on. Okay? However, when you stop burning that energy, let's say you're eating more than you are burning, or you're sitting around not doing anything, then what happens is the citric acid cycle starts to slow down. And as the citric acid cycle starts to slow down, the very first place it will start to slow down is with the molecule called isocitrate. Isocitrate is produced from citrate. Well, if isocitrate starts to accumulate, citrate's going to accumulate. And if citrate accumulates, this is only going to happen when the cell has more energy than what it needs. So citrate, for that reason, is an indicator of energy. And citrate is an indicator of high energy. When citrate accumulates, 2 things can happen. One is it will shut off glycolysis, just like you see right here. Okay? And second is citrate will be used to carry acetates out into the cytoplasm, because the citric acid cycle is occurring in the mitochondrion. So accumulation of citrate in the mitochondrion means that it gets taken out of the mitochondria and put out into the cytoplasm. And in the cytoplasm, it gets broken down into acetyl-CoA and oxaloacetate. Acetyl-CoA in a cell that's full of energy gets made into fatty acids. So some of that doesn't get broken down into oxaloacetate and acetyl-CoA, which means that the cytoplasmic concentration of citrate starts to increase. And that increases when the cell is full of energy. And, of course, in the cytoplasm is where glycolysis and gluconeogenesis are going on. And so when the energy level of the cell is increasing, you don't want to be breaking down glucose. You want to be making glucose, and that's what's happening here. Make sense? Yes? Alanine is a sort of an indicator of high energy, okay? But alanine, more importantly, is an indicator of an abundance of pyruvate, okay? Because pyruvate, it's a one-step reaction to go from pyruvate to alanine. So alanine tells the cell you got plenty of stuff, because it turns out that, okay, you think of the Atkins diet. The Atkins diet says low carbs. You don't need carbs, right? If you don't need carbs, then how do you get all glycolysis and how do you get all this stuff going on, right? Well, the way that you get this going on is through breakdown of amino acids. Breakdown of amino acids can give you the energy that you otherwise would have gotten from glycolysis. And there's a couple ways in which this can happen. You'll see some of those happen with citric acid cycle next term. But one you can see right here. That is, alanine can get converted to pyruvate, and pyruvate can get converted to acetyl-CoA and then oxidized in the citric acid cycle. So you see amino acids that can be used for energy. You don't have to start with glucose as your energy source. Now, more importantly, amino acids like alanine and things like that don't stimulate insulin production. And insulin production is probably something you want to avoid. The more you avoid that, the better off you are. So that's one of the reasons why I think a low-carb diet makes a lot of sense. Okay. More than I was asked, but that's what I'm telling you. So I always tell people more than I'm asked. Opinionated old guy. Okay. Anything else? How does it what? How is the carbonic anhydrase mechanism assisted at higher pH? I probably will give you an unsatisfactory answer to that, but I'll give you an answer nonetheless. How's that? Okay. So carbonic anhydrase reaction is dependent, okay, upon, and I'm not getting my thing here, is dependent upon, okay, the activation of water, okay? Anything that's going to favor the activation of water will favor this reaction. Well, in a high pH, it's much more likely this will lose the proton than in a low pH. And that's why I say that's the unsatisfying part of that answer, is that at a high pH you see ionization of water much more readily, loss of protons much more readily, like you do any acid, than you do at a low pH. And so that's what's actually going on right here. I hear the typing has stopped, so that must mean you've caught up with me finally. Damn it, there I did it again. I got to just shut up. My wife has had a cold, a terrible cold this past week, and I'm telling her, just quit talking. Just quit, and I'll say quit talking, and then 2 minutes later I'll say, so what do you think about such and such? And I'm going, I can't even listen to myself, so. But you know that probably very well. Okay, anything else? Justin?
I just have one quick question. I can't remember which, there's an enzyme within gluconeogenesis that's assisted, or that's carboxylated
So the synthesis of oxaloacetate, his question is there's a molecule that gets carboxylated in gluconeogenesis with the assistance of biotin, and the answer is yes. The molecule is the conversion of pyruvate into oxaloacetate, because that's going from a 3-carbon molecule to a 4-carbon molecule. The enzyme that carries the biotin and that catalyzes that reaction is pyruvate carboxylase. And the biotin has the sole function of carrying the carbon dioxide to the place where the carboxylation reaction occurs.
And I was wondering if that had a role, if biotin had a role in the carboxylation of prothrombin.
Does biotin have a role in the carboxylation of prothrombin? I think somebody actually asked that in the class earlier. As far as I know, I don't know the answer to the question definitively, but I would be amazed if it didn't. Virtually every carboxylation that happens in the cell uses biotin. So it would be unusual for a mechanism to exist that didn't have that. But I don't know that definitively to tell you that. But if I were guessing, that's what I would definitely guess, yes. Biotin is an essential nutrient. What is it, vitamin B6? Is that right? Does anybody know? Nutritionists in here? It's one of the B vitamins. So. You're going to look it up? Yeah. Yes, question. On the what? V versus S, meaning what do you? Yeah, I know what V versus S is, but what are you wanting to know? Okay. Well, if you look at enzyme kinetics, then if you look at enzyme kinetics, what you see is that for most enzymes, not all enzymes, but most enzymes behave in the way that you see here. And what that means is that if you take an enzyme and you take varying amounts of substrate, where you do a set of different reactions, each with a different amount of substrate, and you look at the rate of production of the product, which is what's known as the velocity. If you look at the rate of the formation of the product, what you see is that as you add more substrate, you get more velocity. And that's not totally surprising because it takes substrate to make for the enzyme to make product. So the more substrate it has, the more product it should make. And you see this phenomenon. And though I didn't talk about it here, I'll actually show you here since I'm on this figure. What you can see is that there is almost a linear response very early in the reaction. That's what they're trying to depict here. That's this initial velocity that's occurring inside of cells. And initial velocities, the reason we measure initial velocities are because the accumulation of product starts favoring the reverse reaction. It starts making the reverse reaction more possible. So when we study velocities, we always want to see what initial velocity is. So each of these were studied for very short periods of time so that we didn't have much accumulation of product. And in each case, we were measuring initial velocities in these reactions. Now, so the curve, however, doesn't stay linear. The curve does start to flatten out. And the reason it starts to flatten out is because the enzyme is getting saturated with substrate, so that adding twice as much substrate doesn't have the effect of doubling the rate simply because the enzyme is busy. You know, you've had a busy day where you're doing something and somebody comes and piles twice as much work onto you. It doesn't mean you're going to get twice as much work done. It means you're going to go, my God, I've got too many things to do, right? And you're going to complain, and you're going to be up here in this mode, all right? I'm saturated with work, whereas down here, where you walked in the morning and they gave you this work, you didn't have much to do. They gave you the work, you got it done. Plus, you'd had your coffee and you were cooking, all right? Okay, bad metaphors here, but all right. So late in the day when they've given you all this work, you're up over here. All right, so what that means is that there's really a maximum amount of work, or in this case, a maximum velocity that an enzymatic reaction can achieve, and that velocity is limited by the amount of enzyme. So since we're doing the same amount of enzyme in each one of these reactions, as we add more and more substrate, we are getting to the point where the enzyme can't handle any more any faster. And so we reach what we refer to as maximum velocity, or Vmax. And Vmax occurs when the enzyme is saturated with substrate. And I'm looking for my Vmax. Where's my Vmax? Well, there's Vmax here. Okay, so I don't like this figure because I think these lines are too far apart, but Vmax is the place where the enzyme is saturated with substrate. So that is, Vmax, as I said in class, is a quantity that is dependent on the amount of enzyme. If I double the amount of enzyme, I will double the Vmax. So Vmax doesn't tell us much about an enzyme because it depends on the amount of enzyme. But if I take the Vmax and I divide the Vmax by the concentration of enzyme that I used, the concentrations drop out and I get a number. And that number corresponds to the number of molecules of product per molecule of enzyme per time. And that's a very valuable number. That number is what we call kcat, or the turnover number. They're the same thing. And a kcat of 100 per second means that each molecule of enzyme is making, what did I say, 100, let's say 1,000. A kcat of 1,000 means each molecule of enzyme is turning over or producing 1,000 molecules of product per second. So that's a very useful number. And I can compare kcats of different enzymes because it doesn't have any relationship to concentration of enzyme. The last entity here, of course, is the Km. And as I predicted on the exam, about half the students tried to tell me that Km was the Vmax over 2. Now, you can see Vmax over 2 is right there. You can see Km is right there, so it's kind of hard to argue that they're the same thing. In addition, Vmax is a velocity. Km is a concentration. So it's like saying 30 miles per hour equals $1.99 a gallon. I mean, it doesn't make any sense. So they're not the same thing. Km, however, is the substrate concentration that gives Vmax over 2. And that's the relationship between them. But they're not the same thing. And there were about, I'd say, close to a third of the people on the exam that when I asked them that appropriately, they tried to tell me that that's what it was. So, in fact, I had people, this is a true story, I had people ask for regrades where they tried to tell me that that was the case on the regrade. Yeah. So. Yeah.
Are you still going to have the equations on the final?
The equations will be on the final. I should have said that. People always ask that. Oh, my God, he's giving us a card. Do we have to write the... So say I'm saving you space on your card. I have seen cards that are literally works of art. I mean, some of them are just absolutely incredible. I may have told the story in this class. Did I tell you the story about the person doing the 2 different colored glasses? Oh, God. So this is, you can't do this, by the way. I'm telling people out in TV land this as well. I had a young man who I knew him very well. He'd been in a summer program that I run. He was very creative. And he says, I know how to get twice as much information on a card. And I said, oh, really? He said, yep. He said, can I do it? I said, whatever you want to do, try it, you know. So what he does is he takes his card. I said, you know, you can't paste anything on there. You can't use figures. You got to do it all by hand. He said, it'll all be by hand. It'll all be done, et cetera, et cetera. So what he did was he took his card and he wrote in the tiniest writing that he could in a green pen everything that he wanted on there. And then he took on the same card and with a red pen, he took and he wrote everything on there as small as he could on there. And then he brought to class red and green glasses. So when he looks in one glass, he sees one set of writing, and when he looks in the other, and it worked. And he got twice as much information on his card. But I outlawed it after that. But he got it. But I thought it was a pretty clever idea, you know. He got twice as much information on his card by using color coding, which was kind of cool. So color coding is fine. You just can't use glasses to tell the difference. I mean, if you want to try to figure it out with your own eyes, you can. And a lot of people use color, of course, to help organize things, which is fine as well. But I banned the use of dual-colored glasses to try to figure it out. But it's pretty clever. In fact, he's the young man who, if you've ever walked into the second floor of ALS, you'll see a little sign on the door that says "Not allowed in building," and there's a thing on there, and there's actually a picture that used to be there that got pulled off. Well, that picture was me. Somebody put a picture of me on there because it's supposed to say like "Not allowed in building," you know, radioactive material or dangerous chemicals or blah. So he was the one who did it. He took this picture of me and he put it on, which I thought was the coolest thing. And he puts this picture of me on there. It says "Not allowed in the building." Okay, so there's a funny story that goes with this. So it's kind of, but somebody since has pulled the damn thing off, which I was kind of bummed about. It was on there for many years. So we had a group of freshmen who were taking a class downstairs with one of our professors, okay? And it's the first day of class, the first day of freshmen at OSU, and they're sitting there and they're talking to the professor saying, you know, what their life is like and what their experiences are like and so forth. And one nervous freshman at the very end of class raises his hand and says, "Do you know the guy whose picture is not allowed in this building?" And the professor says, "Yeah, I just saw him." Well, I mean, we shouldn't laugh because there are places, of course, where people aren't supposed to be and so on and so forth. But I always had this vision that one of these days I was going to walk into my building and that the police were going to come and escort me away, you know? It's like, this comes full circle, okay? So it comes back to this, and I'm kind of amused at this and so forth. And so I locked my keys in my office one night. And when you lock your keys in your office, of course, you can't get in the car and go home. And so the only thing you can do is call the cops, right? So I call the cops to come and let me into my office so I can get my keys to get out. And the cop comes and he goes, "You're the guy that's not supposed to be in the building." I said, "Yeah." He thought it was very funny. He'd seen this for a long time and he knew it was a joke, but he made me a little nervous for a while. So it was kind of cute. All right, I think that's enough silliness for the evening. Any other questions? I wish you all the best. I will be in my office the next couple of days, so if you have questions, come and see me. Late in the afternoons, I may do like I did today. I'll go home and probably do some work, so if you're looking for me late in the afternoons, that may not be the best time to find me, but otherwise I should be around and able to answer any questions that you may have. So good luck.